So this is principle of transformation, that is, which is the genetic material, that is search for the genetic material was given by these three scientists. Okay, total there are 1, 2, 3, 4, 5, 6 scientists. They had conducted separate experiments. Okay. Frederick Griffith was the first one to conduct experiment on the mice. Then Avery, mccloed and mccarty, they studied. Okay, they did one experiment. They continued the work of Griffith and they further found something else. Then Hershey and Chase conducted experiment using radioisotope. Okay, radioactively labeled sulfur and radioactively labeled phosphate. Sulfur is present in protein and phosphorus is present in DNA. Phosphate bonds. Sulfur is present in protein. Let's see what the base starting. A brief on transforming principle. First, this described the conversion of non-pathogenic pneumonia bacteria to a virulent strain. We have seen there was a strain called R bacteria and S bacteria. R bacteria was not harmful and S bacteria was harmful. This S bacteria can convert R bacteria from normal to a harmful. The S bacteria, the substance which are present in S bacteria, can convert the R bacteria into virulent type. Actually, it is not harmful, R. But when some substance of S bacteria are grown along with R bacteria, the R bacteria became harmful. Griffith mixed the living non-virulent bacteria with heat inactivated virulent form. What Griffith did, he mixed living non-virulent bacteria, which is non-virulent, R, with heat inactivated virulent bacteria. He inactivated S bacteria, he mixed it with R bacteria and he grew it. What he found that there were some colonies which became virulent. He infected the mice with a mixture and much to his surprise, the mice developed pneumonia and died. Now he infected which mixture? Non-virulent bacteria with heat-killed virulent bacteria. Actually, what initially happened, non-virulent bacteria was injected in mice. The mice remained alive. Virulent bacteria was killed and then injected in mice. The mice was also alive. Why the mice was alive? Because they have inactivated the virulent. But when this inactivated virulent heat killed mixed with non-virulent bacteria and then this mixture when was injected in the mice died. Okay, the mice died. Separately when you inject them, the mice survive. But when you mix the mixture and then inject the mice died. So they isolated the colony of the virulent strain from the mice. When the mice was died, they removed the blood sample and they grow it on the agar plate. They take the blood sample, they dilute and just sprinkle on the agar plate. This virulent bacteria was found. Did they inject any virulent bacteria directly? No. What they had injected is S and R mixture. S was heat killed, but S was heat killed. It was not causing any pneumonia. S was heat killed, R was normal. But when they found, they saw that you get a colony which is virulent. Initially, R is non-virulent, heat killed is non-virulent, mixture is virulent. Okay. So there is something called as transforming principle. This phenomena was observed for the first time, but was confirmed a year later by Dawson and Sia, who were also able to perform this transformation in vitro. Now, this Dawson and Sia is not in detail for you all, but what they did in vitro. What is in vitro? IVF means inside the test tube. What Griffith did? Outside the test tube. On life of someone. Here they didn't take any life, they did it in vitro. That is using some bacteria inside the test tube. James L. Holloway lies the virulent strain. What is lysing? Breaking. The virulent strain, that is bacteria, he broke it open, break it open, filtered the intracellular substrate, removed all the substance, extract, transform the non-virulent strain to a virulent. So that extract which he removed from the virulent bacteria. Okay, from the virulent bacteria, he removed the extract. This extract transformed the R strain into virulent type, like into S strain, you can see. Because R strain is non-virulent, S strain is virulent. Something is there in the cell-free extract that caused this conversion. So some substance is there in the extract that caused the conversion, and they called it as the extract transformed the non-virulent strain to the virulent strain. Next is search for the genetic material start in the mid 19th century. In the 19th century, Griffith experiment was conducted in which year? 1928, no?
Mine was 1930. 1930.
Okay.
1935.
Yeah. Who discovered the principle of inheritance? Mendel. Mendel discovered the principle. Mendel is like a first gear.
Oh, John.
Okay.
Johann.
Yeah, Johann or Johann. Yeah, spelling is same. Starts with J. So he was the first to discover the principle of inheritance. What are factors?
It's chromosomes.
Yeah, genes. Initially, they didn't knew that it was called as genes. Mendel gave the word called as factors. Mendel's laws of inheritance is basis for the researchers on genetic material. So he laid the foundation for genetic work. He started working on genetics, and then other scientists started working with genetics. Okay. It took a long time for the acceptance that DNA is the genetic material. Mendel don't know anything about DNA. He knows only characters are transformed because of some factors. He don't know anything about gene also. Later, it was discovered that DNA is the genetic material. So Griffith experiment of transforming principle. He worked on two strains. One is R strain, the other one is S strain. R strain is rough. You can see they have shown rough. S strain because it is called smooth. Why it is smooth? Because they have polysaccharide coat capsule. On smooth strain, they have polysaccharide coat capsule. So same smooth shiny colonies. That's why it is called S strain. While other consist of rough colonies, which is called as R strain. Difference because of presence of mucous coat. There is a presence of mucous coat which is smooth. Okay, on the S strain. Okay. Now let's come to the Griffith part. Okay, see here what Griffith did. He took the live S strain, which is virulent, and injected in the mice. The mouse died. Then what he did? He took live R strain, which is non-virulent. He injected it in the mouse survived. Okay. Then what he did? He took dead S strain. How the S strain was made dead?
By heating.
By heating. He injected this dead S strains into mouse, the mice alive. And then what he did? He took this S strain, this S strain he took and mixed with R strain. Okay. He took this S strain and mixed with R strain. You can see the mixture. Then he injected the mixture. What happened to the mouse? The mouse dies. What we can see here, the mouse lives when it is heat-killed S strain. The mouse survives when it is R strain. But when mixture is added, the mouse dies. Then what they did, they removed the sample from this mouse, blood sample, and they grew it on the nutrient plate. And what they got, they found S strain bacteria was there. So this R strain was transformed to S strain. That is called as transformation. Okay. Here you can see again same S strain, mouse dies. R strain, mouse lives. Heat killed S strain, mouse lives. Heat kill X strain plus R strain mixed, the mouse dies. Blood samples from the blood mouse contain live virulent S strain. That means the R strain is getting transformed to a S strain. There is something that is transforming the R strain to S strain. Next we have, okay, this was the turning point. Griffith experiment was the turning point. But what he was not able to find is he failed in finding that failed biochemistry. What is biochemistry? Means he didn't know which chemical is causing this transformation. We have studied in biochemistry, biomolecules, sugar, all that, lipids, sugar, proteins, and nucleic acid. So out of these four, he is not sure which is causing this transformation. That's why he failed. Then three scientists, Oswald Avery, Colin McCloed, and mccarty, they went in search of the genetic material. Okay, these three scientists, they started searching for the genetic material. What they did, they used Griffith experiment, and they worked on Griffith experiment, but slightly advanced. Okay, they went in search of the genetic material. Okay? These three scientists, Oswald Avery, Colin McCloud, and mccarty, they went for the search of genetic material. Now this is their experiment. So what is transforming agent? DNA or protein? They just wanted to find what is the transforming agent, whether it is DNA or protein.
So how they knew that DNA is a part of it?
Because we have studied biomolecules. In biomolecules, we know there is lipids, there is sugar, there is protein, and there is nucleic acid. Nucleic acid is of two types. Which are the two types of nucleic acid? RNA and DNA. So they know these biomolecules are there.
So it can be any one of them.
It can be any one out of them. Now okay. So every NS team extracted the purified proteins, DNA, RNA, and other biomolecules from the heat kill S-stream. Extracted them. Okay. They observed that protein digesting enzyme protease and RNA digesting enzyme RNase didn't inhibit, but DNase did. Okay. This I will explain in next picture. Okay. Now this is what they did. This is the heat kill S-strain. This first test tube. What is removed out? They have removed out lipids and sugar. Lipids and sugar. Now, what is left in this is there will be protein, there will be RNA, and there will be DNA. First, they took the sample in one test tube and they added proteinase. They took this sample in one test tube and they added proteinase. What will happen to protein? There will be no protein. Proteinase is an enzyme which digests proteins. Proteinase is an enzyme which digests protein. So in this test tube, there is no protein. But what is present in this test tube? In this test tube, there is no protein. What is left then? See, I'll start again. See, there is lipid removed, sugar removed. Lipids and sugars are removed. In this test tube, there is protein now. There is RNA and DNA. We have studied biomolecules. RNA, DNA, protein. Yeah. Okay. Now they take out some sample from this and took it in one test tube. They add proteinase, no protein. So what is left in this? Which biomolecule is left? Biomolecules I'm talking. DNA and RNA. What is removed is they have removed protein here and they have already removed lipids and sugar. So only DNA and RNA is left here. So when this mixture of S strain heat killed was mixed with R strain, the S strain appears. The S strain appears. So what we can conclude, protein has no part to play in transformation. Even if there is no protein, there will be transformation. Okay. Okay. Then next is they took some sample from this in another test tube. They added RNase. What RNase will do? Remove RNA. RNase is a RNA digesting enzyme. So here RNA is removed. What is left now in this test tube? Protein and DNA. Protein and DNA is left. So when this test tube was mixed with, when this material was mixed with R cells, there was S cell appearing. That means RNA also have no role to play in transformation. There is something else in the cells which is transforming. So they found protein has no role. If I remove protein, the cells are getting transformed. If I remove RNA, the cells are getting transformed. In third test tube, they added DNAs. In third test tube, they added DNAs. There was no DNA. What is left now? Protein and RNA. When this mixture was mixed with R cell, there was no S cell, there was no transformation. So what they can conclude, DNA is the transforming principle or transforming agent. DNA is one which is causing transforming. Because here there is no DNA, that's why there was no transformation. Got now this principle? Got the experiment? Easy. For exam, if it comes, you just draw this, done, and just explain some, or just make a flow chart. Okay, now just to show directly, see here. Okay, see here. The S strain is extract is removed, polysaccharide is destroyed, mouse died. Why the mouse is dying here? S strain is there, but polysaccharide is removed. What is left now? Which makes the mouse to die? DNA. Next test tube, they removed lipids. Lipids is written there. Mouse died. Third, they removed RNA. What will happen to the mouse? Mouse will die. Fourth, they have removed protein. What will happen to the mouse? Mouse dies. Fifth, they have removed DNA. What happens to the mouse? The mouse survives. So this tells that DNA is the genetic material. Okay. So this is what you have to know from the first experiment. Third experiment is slightly advanced. They use radioactive isotopes. Okay, to see whether DNA is transformed. This. So no S strain, no live S strain recovered from here. But here live S strain was recovered. Why no live S strain? There was no transformation, no DNA, no transformation. These are all. These are having all R cells. They are having R cell and S strain extract. So there was no recovery of S strain because there is no transformation. Here there is transformation. The R got transformed to S, and they saw S cell. So we'll stop here. Next time we have to do that Hershey chase.